Whether you regenerate and whether you degenerate, we're staying alive, staying alive. Aging, aging, aging here and there. Aging, 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 aging everywhere. Aging is a song that is universally sung. Aging is a thing you can ignore when you are young. But when you're older and begin to have dementia, you'll think back upon this course and wish you'd paid attention. Aging, aging, aging here and there. He was a Buffalo postdoc, taken from Canada, dropped in the Middle East. He was fighting on arrival. He was fighting for survival. Coding with AAI, AAI, AAI. Okay, this is lecture 11. We're going to talk about the Holy Grail that people since Gilgamesh in the ancient epic, right here in the Middle East, seeking immortal life. Is it a fool's errand? Everybody has failed so far. Even the meditators and the great spiritual monks with their physical and healthy lifestyle, regular sleep, good modest meals, meaning, social connectedness, relatedness. Do they live to 140?
No.
Is it a fool's errand? Okay. Yeah, so let's take a nice deep sigh of relief. And I want to, before we get into maximal lifespan, talk about the final project. Because this course also doesn't have a maximal infinite lifespan. In fact, it's going to end soon. All good things come to an end. Also, all bad things. In fact, everything comes to an end. Hello. And final project. So, final project. The idea of our final project is very simple. It's supposed to. The idea is it's supposed to make you feel creative, open, like smiling. Your heart is smiling. That's the purpose. So you need to choose a question that interests you and answer with the concepts of the course. You do it in pairs. If you want to, you can also do it solo. It's pass-fail. It's aimed for something like 3 pages, but it could be more. In the past, people have done mathematical models, simulations, all kind of textual stuff. Some people did a song and video. And all this stuff that we do in this course. So that's my description of the goal of this, that you have something really interesting and creative that you're interested in, answer with the concept of the course, and produce a 3-page document. The time to give it is, Tomer is going to tell you. Come here. Let's give a warm hand of applause to Tomer Levy, the teaching associate. מה אתה אומר?
Like a month.
אז מה זה אומר, מה התאריך? That's how you do it in science. Everything is... Time, you hand it in by the 19th of February, 2026. Giving you 1 month. Okay, I'm sure there's some questions, so let's have them.
Is there a third assignment?
What?
Is there a third assignment?
Will there be a third exercise?
Yeah.
No. No. What else? Yeah?
What other research except for yourself, these papers should we be looking at as kind of for the background?
Can you repeat the question?
What researchers except for yourself should we be looking at for the background information for our sources?
Oh, you're asking about the sources?
Yeah, what kind of, where should I look?
I think you should start with a question and then use some AI to find some papers. I don't know. I don't know how to answer. Yeah.
Could we get data?
You want to get data? Yeah.
Is there any resource?
Okay. So there's one special flavor of projects that we want to do where we will have more mentoring. So maybe I ask Ron and Liad to come here. A lot of people are interested in longevity. A lot of other people are interested in sustainability and climate. So we decided to explore the interface. So Ron, this is Ron Milo.
So, sometimes they say Weizmann is like a family. So in this case, you really see, often in the research you say, the person where you did your PhD is like your intellectual father. In this case, Uri was my mentor for my PhD. I'm now a professor here. And Liad is my PhD, so we have like a whole family gathering here. And so the lab is really interested in issues related to sustainability. How do we sustain 8 billion people, one Earth? How does all this going to work out in the future? And one important thing in that respect is demographics. And a big thing about demographics is also what would happen with longevity and what would, how fast would we transition towards sustainability or not, what's going to happen. So in some discussions that I had with my intellectual father and also engaged with his
Intellectual grandchild.
we said if anybody is interested, so we're very curious about looking at the environmental impacts of longevity interventions. It could be like direct things that are happening really quick, like let's say if you're taking, Ozempic, that might have an effect also on your longevity. It decreases your appetite right now, so eating less now. Eating has environmental impact, so maybe you're decreasing your footprint right now, but maybe it would have other effects in the longer term, just to put it out there. So if anybody is interested in trying to do their final project in relation to the environmental impact of different interventions, so Liad, myself, Uri are curious about discussing
And then
that.
we'll kind of work together with you.
And the idea is, if anybody's interested, we'll be meeting here. Next week, you have the class 2 to 4. So those interested would come earlier at 1 o'clock. And we continue discussion. It could be then we make, fits pairs. We could make pairs working on different components of that problem. And the like. What did I forget?
It's going to be really fun. It's like a big project, and we're going to divide into like a few parts on how do we module this concept of extending life and the effects on different aspects of sustainability. Can the world support such a thing? And we'll have like a few different ways to answer that question, and it would all combine to hopefully a big answer that might be even like go beyond that discourse.
And then maybe even we'll find out that having aged, wise people in the population has beneficial impacts on solving complicated problems. All right. Thank you very much.
When you're joining a family, everything could happen.
Yeah. Yeah. No guarantees, right? Okay. Mechot kapayim Liron v'Liad. So I'm just wondering if it's not a commitment, but if in principle this topic is interesting for you, raise your hand. I just want to see. Okay, great. We don't want too many people. We don't want too few people. Okay, so that was about the final project. In our course, we're now there will be one more lecture. And we spent with the first third of the course, more or less, talking about the quantitative patterns of aging and about the molecular drivers of aging and about the truck and house model as a conceptual framework. And we talked about the aging across the tree of life in different animals, the genetics of aging. And then we switched to lifestyle factors. We talked about moving, resting, digesting. We talked about exercise, sleep, and nutrition. And we're going to talk about in the next lecture about emotional health, emotional meaning, passion, connection. And how does that affect biology? And how does it affect longevity? And we'll talk about the aging brain and what improves with age. Today I want to talk about, and then we had this thing about drugs. So last time we talked about drugs that extend that compress the health span, right? Make you add years of health. We looked at mice. We looked at steepening interventions that compress the health span, and we ranked the combinations that can extend health span, not stretch it out, more years of sickness, but more years of health. And today I want to talk about this problem of maximal human lifespan. And why is it a problem? Because you know that in the last 200 years, you can ask about your median lifespan. Let's say you made it to age 10, you didn't die in childhood. How long do you have left? That number has increased over the last 200 years. A lot. So let's say if you made it to age 10 in 1800, you would probably make it to age 40. And now you make it to age 10, you'll probably make it to age 80. Okay, so, and absolute lifespan with everything increased by 50 years. Age 30 was the average lifespan, now it's 80. So, and that expansion has been kind of linear. That means every day you gain 6 hours of extra life. Okay? That's the median. But if you look at the maximum living person, it's not like in 1800, the maximum living person was 100, now it's 150. It's not like that. Maximum lifespan hasn't budged. Okay? The Bible says 120, still have 120. And why? What's the difference between the median and the maximum? What things in biology differentially affect the median and the maximum? What can we learn from that about what varies between human beings? What can we learn from that about how we can achieve this dream of Gilgamesh to live longer? What do we need to play? So I want to tell you what parameters we need to tune, and then how scientists and biotech firms and stuff are thinking about that. So I'll tell you some of the most promising and also some science fiction-y things, bordering on the, I don't know, mind-blowing. That's the plan. So, so what I mean by, so there's a nice plot I like that has the Guinness World Record human longest living, usually woman, always woman. And there's a good record from 1960 to 2020. 2020, that's 50 years, right? And you record the age of the maximum living human. Oh, he died. She died. Oop, she died. There's one outlier in France, 122. But it's debated. Maybe there was a cheating. Because when you get to such extreme ages, if you're in a situation where there was some reason in 1860 to cheat about your birth date for some reason, then that creates a huge signal for longevity because it's so rare to reach. I'll show you how rare it is. So it's not clear, but not rare. And it's rose in these 50 years by about 0.1 year per decade. And that is because in those ancient times, this, if you think 100, so, right, this is 108, 117, something like that. because these people were born in 1850, and between 1850 and 1900, there were massive changes in the world that caused a lot of improvements in lifestyle, you can see. So we can ask, one thing we can ask is, what parameter has changed historically 100 years ago to allow this slow growth? But it's much slower, as I said, than the median lifespan that over this time increased by 15 years.
The number of people don't expand it?
Okay, good point. So is it the number of people? So the answer is, so of course, how can we answer all these things? Because we have our garbage and truck model, right? And even without it, we know that the risk of death as a function of age rises exponentially. And then kind of slows down around age 90. It turns out that the max lifespan, if you calculate, goes like, not like the log of the Earth population, but the log of the log. What does that mean? That means in order to gain 1 year of maximum lifespan, you have to, in the parameters where we are right now, around 10 billion people, you need to increase the world population by 10. Now, over this lifetime, over this 50 years, world population didn't increase by 10. So, and this is more than 1 year. So it's not population growth. Even though the more people you have, by chance you'll have someone living longer, right? But it goes really, really, really slow with the population. Or you can say that survival goes like e to the minus alpha t. So what is it?
Modern medicine?
Yeah. So I'm asking like rhetorically. You don't have to answer really. This is a question that Ben Shenhal in our group was asking, and I really would like to erase the board. So maybe our trusty teaching associate. Yeah. Right. So I'm going to erase the final project here. Okay. Do you need it? Get it? So. I'll put it like this. If we look at survival to extreme long ages, so I'm going to put 100 years here. Let's say about 1% of the people that get to 100. Okay, so I'm going to put this as 1. This is everyone who made it to 100. Now I'm going to ask, what's their chances to make it to 105, 110? It drops so quickly that I have to put a log here. And to reach 110, it's 1 in 1,000 people reach to 100, reach to 110. And it drops like this. It goes like the survival goes very, very fast. It's not exponential, which would look like this. Okay, so we're talking now in this regime here. So you can ask, still there was some parameter, that's changing over time. So what could it be? So what parameters might change over time? We have the production rate of houses. We have the number of trucks. We have the threshold for death. And we have the noise. It turns out that if I improve the rate of production of houses by a tiny amount, just a few percent, or I should put it like this. If I have a distribution of parameters in the population and ask how broad it is, then even a 5% difference between people in the rate of house production I would see people who live to 140. Same thing with trucks. So these can't be varying in the human population. At least you can't have too many people with much slower houses or much faster trucks. Otherwise, you'll see people live to 140. But it's possible to have quite big variations in the threshold and in the noise, even 20%. And then you basically, they don't affect this curve. They just fall on the data, this variation in the population. Okay, maybe I should say we know that there are heritable factors because identical twins lifespan has a correlation of about 0.5. So the heritability of lifespan is about 0.5. So things like genes and environment are shared between people, but it can't be that those twins that live longer have a slower house production or more trucks. Because if there was that much variation, we would see people living to 140. And we already saw in this course that things like exercise affects the threshold. Remember, we know that because the survival curve of exercisers increases median but not maximum lifespan. It's steeper in an X-critical sort of way. And also, these are nuns and monks which have regular lifespan. They have lower noise. This is exercise affects X-critical. And if you look historically, what happened to survival curves, historically there was a lot of early deaths, and this part is kind of more or less preserved. So this early deaths are what went down historically. But then the next parameter that changes is threshold. So what the model shows is that this slow rise is consistent with the change in threshold between 1850 and 1900, where the threshold went up. Why did the threshold went up? The threshold is how much damage you can stand before you die. That's affected by things like pollution, toxins that give you a steady state amount of damage, in addition to the aging damage, so you die younger. Things like medical care, so that if you have antibiotics or if you have even an infusion of saline solution, it can make you survive a typhus or a cholera that you would otherwise die. That's increasing X-critical. So something happened that increases the threshold in that ancient time, not ancient time, 1850 to 1900. Today we see it as a sharp rise, a large rise in the median, and a tiny rise in the maximum. So that's what basically lifestyle factors in medicine can do right now. So that's not the solution if we want to extend maximum lifespan. Another way to say it is that a Japanese group looked at a large database and gave people score for their lifestyle, exercise, nutrition, and also education and a socioeconomic factor, everything we know that extends lifespan. At age 40, so you have between 1 and 7 points. At age 40, if you have 7 points compared to none, that's a gain of, at age 40, 6 years in your remaining lifespan. But if you make it to age 100 and you have those 7 points, it's only a 1-year gain. So if you're 100, it'll give you a 1 extra year, basically, all the exercise and all that stuff. So that's like, it's not extending the maximum lifespan. So when we say do aerobic exercise and anaerobic and strength and stability, that's good for your health, for your mood, for your strength, for your median lifespan, but not for the maximum. So what can we do? It's a rhetorical question, right? Any questions so far? Yeah.
What about animal models?
Oh, animal models are really different. So in mice, it's not hard in the lab to extend maximal lifespan by 50%. In worms, by a factor of 2. We saw some things last time. For example, the mice that had a growth factor made in their liver that made their blood vessels healthier and they lost less capillaries with age. So more of the roads for the trucks. Their survival curve shifted, just like as if you increase the trucks, because you have more roads. And they lived 48% longer, 50% longer. So you can do that. So why can you do it in mice and not, I mean, what's the difference? Is there a difference between mice and humans? Right, that's a big question also in the field. Also, of course, we know that mice and whales, there's a 100-fold increase in lifespan between 2 years and 200 years. So how did natural selection do it? We saw that in the tree of life. And all these things together, what I just told you, how mice can extend lifespan and maximal lifespan in mice, and how natural selection extended maximal lifespan, and what we already know from the SR model, everything points to the same conclusion. It's not going to help you to change the threshold or the noise. It's going to help you to change the houses or the trucks. So we have to reduce the rate of building new houses or the toxicity per house, and or make more trucks. Or make the trucks better, give them more roads. And now we can discuss how to do that. So I'm going to just write it down. To substantially extend maximal lifespan, we need 1, reduce the house production rate. This is supposed to be a crane. And toxicity, the amount of garbage they make. And 2, increase the trucks that remove the garbage. Okay. Evolution, growing from mouse to whale in about 10 million generations, by the way. So 65 million years ago, there was a little mammal that probably looked like a rabbit, and it was living in the it was eating carrots, living in and able to control its own body temperature. But then there was dinosaurs, and it was very minor. Then there was an asteroid hit the Earth, created a kind of perpetual winter. The dinosaurs couldn't control their temperature. All the plants died. They were wiped out, but the little rabbit survived and started multiplying. And it had all the world full of empty niches. It had the grasslands, it had the sea, it had the mountain. And amazingly, our common ancestors started to diverge. So for instance, this bone structure here turned out to be very good for extending and making a fin. That's for the cetaceans, the whales. Or these fingers could fuse and make a hoof. That's for the horses and the cows. Or these fingers could extend and give you a wing. That's the bats. And this happened within 10 million generations, which is amazing because if you try to do a genetic algorithm to produce an and an OR gate with 20 inputs, you won't make it in a million generations of simulations. So it's like amazing. Anyway, that's what happened in evolution. So how did lifespan changed? So factor 100 in lifespan by lowering house production. How did it do that? By improved, basically DNA repair, such as double-stranded break repair, where mouse is worse than human, worse than whale, and other changes. Each species got to that in a different way, but they converged in the same solution. Yeah.
If you improve DNA repair, wouldn't that refer to the garbage instead of the houses?
Yeah, so if you remember, our model right now for houses are stem cells that become epigenetically modified. they produced differentiated cells that are damaged. And if you can repair them, you're okay. Here's where DNA repair works. But if you can't, they become senescent cells, inflammatory senescent cells.
So you prevent them from making the garbage.
You prevent them from, yeah,
If
right.
You prevent the senescence.
Correct. You prevent them from making the garbage. So what it is, I think you're right. The houses are making less garbage. That's what you mean. Yeah, got it. That's what you mean. I didn't understand your question. They're working at this stage. Okay. Here are the trucks, the macrophages, and the NK cells, which I always draw as a little assassin because they're really good at killing cells by injecting poison, and that's the nucleus. I was once in a course on graphics on cuteness in science. Like it's okay to do cute things because in biology, a lot of times, it's a story. We borrow the human narrative ability to make stories with a protagonist, antagonist. Even we say this antagonizes the GLP-1 receptor, right? It comes from the Greek for antagonist. So anyway, so evolution seems to play here. Also, all these epigenetics are slower the longer the lifespan. So we see in long-lived animals that epigenetic changes, also DNA mutations, also translational errors, the longer their go is 1 over the lifespan, basically. So there's something also evolved to build the houses slower. And so that's how evolution does it. Yeah.
Did someone ever try to do like some sort of unnatural selections? For example, in mice taking the one that lives longer and then taking their offspring and then breed them and so on and so
Yeah.
forth.
So you're asking about, this is called breeding. Yeah. So indeed there's many cases of breeding, and there's experiments on breeding for long life. So one of the classic ones is in fruit flies, where they took the last egg that was laid, hatched it, the last egg, and you got flies that reproduce later and later and live longer and longer. It's still going on, actually. It's like been going on for decades, more and more and more. And I don't know what, they tripled the lifespan or something like that. And also in mice, there's long-lived and short-lived ones. So that works. Yeah. So of course it has worked somehow in evolution, right?
Yeah.
But still, usually when you look, there's like millions of DNA changes that are fixed between different species, and so it's hard to understand exactly which is which. But this comparative aspect is very, very interesting. Okay, so we need to. So we're now. What I'm asking now is how we can change eta and beta. So just things we already know, and we also know that. Wait, let me see. This is the record. Remaining lifespan. Yeah, there was something. There was like a little mystery in our field of aging, and there's papers in Science magazine and stuff that keep saying yes, no, yes, no. This is called the slowdown of hazard, right? But there is a question, since the data is so poor, above 105, we don't have a lot of data, whether there's a plateau or whether it keeps rising. Is it flat or does it keep rising, the risk of death? So flat means you get 50% each year, 50%, you live, die, 50% live. So if you get to 110, you have a 50% chance to get to 111. Or your remaining lifespan, let's say, is whatever, 2 years. And when you get to 101, it's 11, it's still 2 years. We have 12, it's still, because that's what means constant hazard. Rising hazard means the older you are, your remaining lifespan also is going down because the risk of death is getting higher and higher. So it's not clear. It's not clear. Some people say this, some people say that. But our model has a mathematical prediction that it keeps rising, like t squared. So I think that or rather, remaining lifespan will keep decreasing. And I think that in the next, 10 years, probably there's so many more people reaching 100 now that we'll have good data and we'll be able to tell one way or the other. Okay, so what else do we know? We know that it's easy to to build more houses, which is a catastrophe, because of these rare diseases of accelerated aging. And they have to do with DNA repair of a particular kind that that has to do with somewhere in this regime. So this RECQ helicases. We also know that early menopause has less severe variants. Same thing. So we have a convergence of two human kind of factors. Same RECQ. It's really interesting. So we know how to screw up things by making more houses. But as I say, it looks like it's very rare to have the opposite, like a person with very low houses. And why that is a mystery to me. I mean, either there's some biological limit, I don't know why there should be. It might also be this saying we have in biology that it's easier to destroy than to build. So it takes a skilled engineer to build a cabin, but any mule can knock it down. Or in other words, if this repair requires a lot of, if improving the repair means improving a lot of things, you need several mutations at once, which is much more rare than a mutation that destroys a part. So if it's like an AND gate over many things to improve, that's one way. But I don't know why. I don't know why we don't have mutation, much more people that live to 140 like that. I mean, it's a mystery to me. About the trucks, that's more clear. I think that more trucks would lead to more autoimmune attack. So that's, if you think these trucks are driving through the city and clearing out the garbage, and sometimes they're colliding into buildings and breaking things and making noise and stuff. More trucks create collateral damage, and there are diseases where you have excess macrophages. You get these cytokine storms, and they're very, very bad. So that's maybe there's a limit to, like a balance between benefit and cost of the immune system. So we need to, so I would say, in order to extend max lifespan, we need to make a radical change to human biology. Then we need to intervene here and here. And that's what I want to tell you, how people are thinking about that. Any questions at this point?
If you would take a human being from, say, 200 generations ago, and you would give them like modern
Medicine
and modern like pollution-free lifestyle, would they live to 80 or would they live to 60? Like, did we have change during the?
Okay. So what you're asking is, did our genes change over 200 generations?
Yeah, it could be.
Yeah,
we
genetics.
grew them.
Yeah. So the timescale of genetic changes in humans is on the order of 1,000 years. And we know that because of amazing work on lactose intolerance. So when babies drink milk, and they have a gene that cuts the milk sugar, but that we're programmed so that goes away when you grow up. But most of us can eat cheese and milk, and the reason is that once there was farming, there were these societies where if they could drink milk as adults, they get extra calories and survive, and that is enough fitness selection that those rare individuals that had a mutation allowing them to digest milk as adults basically grew exponentially and took over the world. Some people even think that's part of those huge Indo-European language transitions. So that's order of thousands of years for fixing a gene like that. So I think people 200 years ago were genetically identical to us.
200
But
generations ago.
200 generations ago means 4,000 years, right?
Yeah.
So most of them, I think it depends where they live, but maybe they couldn't digest lactose. But to think that they would have a different lifespan, I think I doubt it. But I don't know. That's a good question. Okay, so one way, looking back at now it's also I'm kind of reviewing our course. We had a course where we learned from yeast what the driver of the houses was, and it turned out to be a modification on the histones, the proteins that wrap the DNA, some acetylations and stuff that accumulate. So the most elegant thing was if we can reset those epigenetic modifications on histones. That will be, as far as we know, dealing with the cranes building the houses. So we can reset histone modifications. And amazingly, biology takes great care to actually do that every generation. Because there is one example of immortality, you might say. That's called the immortal germline. And what do I mean by that? It's like along our lineage. You take a 40-year-old egg from a 40-year-old woman and a 40-year-old sperm from a 40-year-old man, and they fuse, and they make a baby. How old is the baby? 0. All those 40 years of epigenetic stuff is erased. If the age of that baby was epsilon, then over 1 billion years, the baby would be very, very old. The species wouldn't survive. So our lineage goes back to the first eukaryote 1 billion years ago. And so if babies get older and older, it's by a tiny fraction, non-measurable. So there's something resetting. And when biologists look what happens, what's reset when a sperm and egg fuse, they find a process that's in fact conserved also to mice, also to worms, even to yeast mating. And that process is called epigenetic reprogramming. Resets the histone marks. And the way it works is like this. In the sperm, so the DNA is wrapped around these histones. The sperms do it really dramatically. They throw away the histones and they replace them with a different protein called the protamine. And their DNA is really, really condensed inside their head. They're basically like a warhead with condensed DNA. And they throw away all their old histone marks. Elegant, right? Do you know that? Who studied biology here? Did you know about that, protamines? No. And then you have the egg. The DNA is wrapped around the histones with all their acetylation marks, etc. And the egg has this wave of enzymes, the histone deacetylases, that just clean it, and all these enzymes. So they get basically clean set of histones in most places. There's a tiny bit of information that's passed that's important for development, but in general, there's this resetting wave. And then after they fuse, there's another resetting, and then as you reach the gastrulation stage, that's another. There's these waves of epigenetic resetting. So scientists said, wow, let's try to mimic that process in the adult. And now we're going to take a break. Aging and genetics, they go hand in hand. 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 We'll learn from yeast, that magical beast, how aging and genetics, they go hand in hand. Some people live for 100 years. Some people live for 100 years. They smoke their cigars and they drink their beers, and still they live for 100 years. How do they smoke and still not choke? 'Cause aging and genetics, they go hand in hand. Yeah, aging and genetics, they go hand in hand. 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 Aging and epigenetics, they go hand in hand. Partial reprogramming is where we stand. All right, so we saw that the egg and sperm take a lot of effort to erase their histone marks in order to produce a 0-year-old embryo. And that is, as I said, even conserved to yeast. Yeast mother cell produces a daughter cell. The yeast mother cell has a chromosome. It kind of duplicates the chromosome, and the chromosome on these little rails goes to the daughter cell. And on the way, on the bud neck, there is a histone deacetylase that erases all the histone marks. And then you can actually see it, that the chromosome in the daughter cell is compact, and the chromosome left in the mother cell is more open. That's what happens with aging. The chromatin opens. In a certain sense, the mother yeast is more wise, because the mother yeast, the DNA is open because it's open in genes the mother cell has learned are important to survive in this particular environment. The daughter cell is all compact. It's all plastic and new. It's a generalist. It can learn new things. The mother cell is a specialist. It's already programmed for the particular history that the mother cell saw. That's like a different animal almost. It's old and young. But anyway, it's the same histone deacetylases, basically, that happen all the way up to humans that do the same thing. That's pretty amazing. Also, the mother cell also gives the daughter cell fresh lysosomes, fresh mitochondria, keeps the damaged ones. And when the yeast mate, they can also mate. It's another way of reproducing. There's pheromones, they fuse, they make 4 little spores. And in the middle, they make another garbage can with all the old lysosomes and nuclear membranes there that they eject the garbage. It's incredible. That's in yeast meiosis. So anyway, this is a fascinating field of research. It's the one immortality that we are certain of. And so scientists say, okay, let's try to recapitulate that. And how to do that? So the first step was a scientist called Yamanaka. He won the Nobel Prize in 2006 by discovering that it only takes introducing 4 proteins into the cell in order to take adult cell. Let's say take a skin cell. And you add 4 factors, the Yamanaka factors. They have names like OCT4, SOX2, KLF4, and MYC, which are transcription factors, things that reset the chromatin, methylation, etc., etc. 4 factors. And you get back to embryonic stem cell, which is basically, you can take an adult cell and go back to making a baby that's genetically identical. You can say that. You can take that cell and now cause it in the dish to become neuron cells. So if you take a cell from somebody who's diagnosed with mental illness, you can make neurons and study how they're different from a person without mental illness. This is a big revolution, iPSC. pluripotent stem cells. So that's from the adult cell going all the way back to embryonic state. That's called reprogramming. So in 2016, the lab of Belmonte said, let's take a mouse and induce genetically these 4 factors inside the mouse cells, the living mouse cells. If you do that full strength, all the cells become embryonic stem cells, and then they become a horrible kind of cancer called teratoma. Teratoma is a cancer that grows teeth, hair, intestines. It's a pluripotent. Horrible, horrible cancer. It exists rarely in humans. So you don't want to reprogram all the way back, because if you go all the way back, you go back to an infinite potential cell that can become anything else. It quickly becomes cancerous. So instead, they did a lot of experiments. They pulsed it 2 days every week. And to make the experiment fast, they took progeria mice, which live very short, a few weeks. And they did that, and the mouse lived longer. And because of the shape of the curves, it's scaling, which means they slowed down the houses, which is just what we would predict. So that was a revolutionary paper, and lots of companies were formed, including Altos, which was Amazon company. That takes fantastic scientists, gives them a lot of money, and converts them into people who don't publish anymore. Based on the idea of partial reprogramming. I don't know, maybe it'll work, but it's black box behind the paywall, and you can't tell. There's another impressive experiment by the Sinclair lab where they took the poor mice, they crushed the optical nerve, so now they're blind. And they did partial reprogramming, and the mice could see. So this, again, led to a biotech company that's developing this now for a kind of blindness that happens in similar degenerative blindness. The other companies are trying to take these 4 factors and, for example, delete out MYC. MYC is a well-known cancer gene, so only 3 is enough. And then there's other kinds of factors trying combinatorially many different things. So it's quite a large industry. And of course, that's I think the state of the field right now. Questions? Yeah.
Regarding the histones, isn't it best for like a creature to leave its histones as is from when it becomes an adult? Why does it change?
Can you repeat the question?
I don't understand why evolutionary the histones need to change after an organism reaches maturity.
Why do the histones accumulate marks with aging?
Yeah.
So it's not exactly your part of it is designed to open up the DNA and close the DNA in order to achieve some functions. That's called plasticity. So you have the same genes, but the organism can be different according to the history and the environment. That's called epigenetic changes, and that happens sometimes only in youth, but sometimes also in the adult. So a gene is closed up, but now there's a situation where you need to read it. You open up that DNA. Other things happen by mistake, or you can't avoid them. For example, when there's DNA damage, the repair crew has to work, so they move all the histones and they repair. And in that process, the histones don't come back to the same configuration as before. So it leaves a scar. And that process is a stochastic thing. So almost every cell generation, there's a double-stranded break somewhere. And that causes random scarring. So that's accumulating with time. There's also, inside closed chromatin, there are rare events like an acetylation of the histone because of some chemical acetate or something in the metabolism of the cell. It just happens entropically, and since it's closed, it's hard to remove it. And so all these processes. But you can evolve kind of repair mechanisms, but the pressure to really reduce that rate has to pay off evolutionarily. So a mouse, it doesn't pay to reduce that rate so the mouse lives 100 years, because the mouse is going to live 8 months in the wild. So as I said, to build a machine requires a skilled architect. So that's evolutionary currency. You need selection pressure to build a good machine. If you don't have the selection, every mule can knock it down. Quickly mutations accumulate, and you get to a minimum level of function needed for the organism's optimal fitness in their niche. It's amazing, yeah.
Did people try putting HVAC directly into cells and see what happens?
Yeah, for sure. So this puts the, here you can put histone deacetylases or the RNA that makes the histone deacetylases. So I would say one cutting edge approach called gene editing aims to make design changes. So for example, I'll give you an example. Suppose you have H4K16 acetylation happening in some virus, some repetitive gene that's causing this to open up, and then an ancient virus to jump and cause inflammation and senescence and stuff like that. Gene editing means you basically take something like the COVID vaccine, which is a little oily droplet with some RNA in it that enters the cell, because the cell membrane is also oily. That RNA makes a protein. Now, what do you encode here is a protein that knows how to bind particularly this repetitive DNA. It's a kind of a CRISPR-Cas9 kind of protein, and it's linked to one of those histone deacetylases. So you localize this in order to get rid of this. That's a kind of a strategy people are thinking. And then you can edit the epigenome. You can edit the histone marks in a location-specific manner. Now, because of COVID vaccine, this technology has become so much more powerful that the idea of gene editing is growing very rapidly. So there's one use for this is for very, very rare genetic diseases. So if you have a genetic disease like cystic fibrosis, that's a change of one letter in your DNA, you can use this to put in gene editing that, instead of removing histone acetylation, basically replaces the gene with the correct gene. And now it's a cure. It's a one-shot cure for these rare genetic diseases. I think there's 12 rare genetic diseases that have been cured like this so far. That's a huge revolution. It's actually in gene editing inside the human. And it's just the beginning of it. So these lipid nanoparticles are getting better. The RNA, everything is getting better here. It's like an engineering thing. Editing our own genes. Like people think maybe we should edit the genes where if they're mutated, they give you leukemia. You should change that letter or whatever. So it's like, who knows where this will go. But from the point of view of EDA, this is like, I just wanted to give you an example of something that's in principle possible to do. If you're interested in doing this experiment, talk to me, like Yam. In human blood, I mean. Okay, so now I'm getting into the cutting edge. So we said we have partial reprogramming, we have the ability to edit genes and maybe chromatin modifications. So we're getting into here, edit, reprogram. And maybe we can also enhance DNA repair by adding like whale double-stranded break repair proteins into the human cells. That's one direction. The other direction is in the trucks. So there's ways to take cells out of our body, give them receptors that detect senescent cells, and put them back in the body. It's called CAR T cells. By the way, improving the trucks is borrowing technology that was used to fight cancer. So cancer is the flagship of biology right now. All the new techniques are developed in cancer, the drugs, the billion-dollar drugs there. The fraction devoted to aging research is very, very tiny. It's less than the probability of reaching age 117 if you made it to age 100. Out of the money spent on it. But still, this technology exists, so these CAR T cells that are programmed to kill mutant cancerous B cells are used to kill zombie cells. That worked in mice. Another way to kill zombie cells is to inhibit their anti-suicide protein, BCL2. So they are damaged and they don't kill themselves, but if you inhibit that, you kill senescent cells. That's called senolytic. And that works it was developed for cancer, but it can't be used in humans because it causes side effect of reduced platelets. So clinical trials now isolated, like in the eye, human eye glaucoma, diabetic glaucoma, the first clinical trial with senolytics showed some promising results. Because if you use it in the eye, it doesn't go in the whole body and doesn't cause those side effects. And here in Weizmann, there's, I think, the most exciting direction is discovered that just like cancer cells, also senolytic cells have a sign that says, "Don't kill me," to the immune system. It's called PDL1. And if you put an antibody that blocks the sign, the trucks kill senescent cells more. That's called checkpoint inhibitor. And that's used to kill cancer. That's a revolution in cancer because up to 10 years ago or 15 years ago, if you had stage 4 cancer with metastases all over your body, it's a death sentence and virtually nobody survived more than a few years. But this checkpoint inhibitor, about 20% of those people can survive for a long time. So that's like a huge revolution in cancer by unleashing the immune system. The side effects are autoimmune diseases. So what's amazing about killing senescent cells is it's much easier to do than killing cancer cells. Cancer cells divide and mutate, so they're very sneaky, and if you try to kill them, they'll find a way to resist it. And if you want to have the immune system kill them, you need a massive checkpoint inhibition. Senescent cells don't divide by definition, and they don't mutate enough to avoid killing. They don't. It's not like they divide, and you have some mutation. That's one who's resistant, and it's not going to be killed, so it's going to grow exponentially and restore the cancer. It's not like that. So I think it's going to be much easier to destroy senescent cells. I'm kind of optimistic about that. They're an easier target, and there's multiple ways to do that. And there's evidence now in mice that PD-L1 checkpoint inhibition also works for Alzheimer by destroying senescent glia, the trucks of the brain, by unleashing the peripheral immune system to go into the brain. That's Michal Schwartz's work, and this work by Valery Krzyżanowski, who also participated in this senolytic. So Weizmann is a big center for this senolytic field. So these approaches, you see, there's multiple ways forward here to improve the houses, and to reduce houses and improve trucks. Okay. And I'm going to go into more like futuristic things or things that are happening right now. In order to develop a drug like that, so it works in mice. Now you want to try it in humans. So PD-L1 checkpoint inhibitor is already approved in humans, so that's a good start. Even though for killing senescent cells, you can even use a short-lived antibody that doesn't interact with the immune system. It's less inflammatory. It's probably even better. But once you have a new drug, it usually takes 10 years and $1 billion to get it to be a drug for humans, for mice to humans, and 9 out of 10 times that fails. It fails because efficacy, because mice are different from humans, the drug doesn't work, or toxicity, it has a side effect that you didn't see in the animals. That's a major bottleneck, right? If you can improve the drug design pipeline by a few percent, it's worth a lot of money, right? And the big hype right now, or hope, is that artificial intelligence can improve all parts of that pipeline. And I used to be skeptical, but I'm now not so skeptical because there's one aspect that was revolutionized by having a large training set and a good learning algorithm, and that's the ability to predict how a sequence of amino acids, a gene, translated gene, would fold in 3D and make a protein. That problem, when I was a postdoc in 1995, 6, 7, 8, was like we knew that whoever cracks this problem gets a Nobel Prize. And the idea is to find the chemical push and pull of each of these amino acids on itself to find the three-dimensional fold. And there's a lot of like crystal structure data sets that grew. And a few years ago, AlphaFold, the software, it became so good at it that experts were like, and basically it changed the way you look for drugs, because now you see the gene, you know how this 3D structure, and then you can design the lock and key molecule to bind it. So that's a big step forward, and it's anticipated that other parts of the drug pipeline will also be improved. One big is target. What part of the cell to target? So we say H4K16 acetylation and line 1 elements. That's a target. So whether artificial intelligence can help you find targets, I don't think so. I think it's human intelligence with artificial intelligence as an engine. Next, toxicity is this. So you have chemical space, so you can encode a foundational model based on chemical space, graph neural networks. So in chemical space of small molecules, weigh like 500 hydrogen atoms, so a few carbons, which one of them is toxic to the human being? So you can screen them against all proteins and see if they bind. Which one of them is going to enter the intestine, cross the blood-brain barrier? All these questions maybe are solvable. This is a huge game. All these problems, anyway, are large research fields. So some companies now are accelerating the rate of drugs brought to clinic, and I think probably in 10 years we'll see a big speed up. I don't know exactly. Questions about that? No? Do you have a question? 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 You? Yeah, you have a question.
How do the telomeres get into the whole story?
Telomeres, the ends of chromosomes, it's the same thing. The histone acetylations open up and remove this protective shield, and then there's double-stranded breaks here, and then the telomeres start fraying and shortening. So I would say it's a side effect of the problem of the same building of houses. And in most cell types in humans, it's not causing aging, but it's associated with the same problem. And what causes aging is probably the same thing, opening up viruses that are jumping and making the cell think it's inflamed, become senescent. Double-strand breaks also in the telomeres in repetitive regions that can't be repaired, making the cell think it's senescent, things like that. Just extending telomere is not going to make you live longer. It's going to give you several kinds of cancer. Okay, so there's an idea called, this is just like philosophy, it's called longevity escape velocity. Did everybody ever hear about that? Yeah? So escape velocity, right? If you go fast enough, you can escape the orbit of the Earth, like rockets. So if we know that lifespan right now is growing 6 hours every day for the last 200 years, and we get better and better at extending lifespan, maybe there'll be a day where lifespan increases by 24 hours every day. And then you've reached longevity escape velocity. So just wait and hold on. It's an interesting idea. I talked about gene editing. There's a whole field about blood. So blood is like so it's like, vampire drinks blood to live forever, like David Bowie in the movie Hunger. But so it's a very attractive idea, but I think a lot of that work is not reproducible, hasn't been reproduced. So you take blood from young mice, you put them into old mice, or you even purify some factor. But some companies used to sell serum from young people for $8,000 a liter. Put it into, but it's so far not reproducible. So I just want to tell you about that. Then there's the whole thing about engineering technology, not biotechnology. So there's whole fields trying to combat fragility. So the one field is exoskeletons, which are light materials with motors that just help you with your muscle fragility. So you walk around, you hike, and you fall less. What do you think about that?
How did that happen again?
You have a machine, external skeleton, made of rods and motors, and AI.
Well, it effectively acts as if you exercised all your life.
Yeah,
So it just raises X critical, basically.
it's like raising X critical, in a certain way. Okay, right. It's not a lifestyle. Then there's companion robots that are gonna help you with loneliness and social isolation. That we'll talk about next week. And then there's advances in the ability to deal with mental illness by deep brain stimulation. So you put an electrode or externally able to read. This is from surgery on people with epilepsy. You have to do awake surgery and find out, okay, if I zap you here, oh, I see red. I zap you here, I smell an orange. So in order to know exactly where to intervene to stop the epilepsy and not remove your functions. So researchers use that to record, and with machine learning, you can kind of understand the language of the brain, and then you can inject currents and control the brain. Control the brain, like reduce OCD, that works. So there's something about that I find chilling, and they're thinking that at some point there'll be a kind of feedback loop that maybe will help with neurodegeneration and memory and stuff like that. And then there's all the other feedback loops in our body that we can think of helping. So an example, people with type 1 diabetes, they lost their beta cells and they can't make insulin. So in modern world, they, instead of injecting insulin manually, measuring blood glucose and injecting insulin, they're wearing a pump and a glucose sensor, and the glucose sensor is measuring their glucose continuously, and there's a feedback loop with the pump secreting insulin. It's not such an easy problem. It's only recently been solved in an okay way. So this person is walking around with a machine that's basically closing a control loop on their glucose insulin circuit. And in the same way, you can imagine doing the same thing for all other control loops, like all other important hormones, sex hormones, and stress hormones, and other important metabolites. All you need is a pump and a sensor and a good understanding of the logic. Again, I think you're right. It's increasing X critical. So maybe I should put this in the increasing X critical file. And then there's another idea that's science fiction-y, which means that after we reach longevity escape velocity, if we hang on long enough, one day we'll be able to download our brain into a software. And that makes me uneasy. I don't know what makes me more uneasy. It's the technical challenge or the weirdness of transferring your intelligence to a piece of silicon. Like, what's this philosophical question of continuity, right? When I wake up, I'm sure I'm Uri for some reason. But if I wake up now, I'm like on the cluster here at White. It's different. But amazingly, there are companies that are pursuing all kind of brain-computer interfaces with all kind of ideas, and it's not impossible that in 50 years or something it will be some part of that would be something you can take seriously. What do you guys think? Yeah.
Don't they also have like all the, printing new organs and that thing?
Oh, printing new organs.
Yeah.
That's another science fiction-y thing, right? Printing new organs. There's also humanization. So you can take pig heart and humanize it by gene editing so that it doesn't cause immune attack, so that you get a large source of organs. So yeah, that's an approach. Maybe I should add it. This is called, it's like you keep replacing organs.
Wouldn't that all just, but doesn't it seem like every organ you introduce is just raising X critical again?
I think so, yeah.
Like you're not actually combating the whole
Yeah.
system falling apart.
Yeah, unless you can find out where those stem cells.
Blood vessels,
Yeah.
that's fine.
I think, yeah, actually, so our conversation with the SR model is, first of all, it's unique to this classroom. It's not that everyone in the aging field talks about X critical and houses. It's just very confused discussion. So you lump all these things together and you don't know what they do. But I think what you're saying here is true. Some of these are more X critical, some of them are EDA, and some of them are. So that's a very important distinction. And so if you keep changing organs and there's some part in the body that keeps making zombie cells, it's going to wear out, something like that. And the brain also has its own clock. Anyway, I think out of all these solutions, it seems to me that for my favorite is senolytics right now. And I think once that works, we'll probably expose the next layer in the onion. Maybe brain related lysosome, mitochondria, protein aggregation problem. And then we have to think again. So hopefully there's a finite number of layers in the onion. And also remember the idea from last time that if you reduce EDA, you stretch time like a rubber band, and therefore likely increase the six span. So you also need to do both of these. But beta, it's good because it's steepening and it should extend lifespan, but shrink the six span. So that's another consideration that's unique to you guys. I give it to you as a gift of knowledge. Yeah.
So do people that currently take checkpoint inhibitors for beta-1
Yeah.
have lower senescence and live longer other than the cancer?
I doubt it because the question is people now that take PD-L1 for cancer. The treatment PD-L1 for cancer has a lot of serious side effects. I mean, tens of % of them have an unleashed autoimmune attack. The autoimmune attack could be thyroid, it could be parathyroid, it could be all kind of. In order to combat this, you need steroids. So they take glucocorticosteroid that causes a whole other range of problems. So right now, it's not the final word on it's for cancer, but the drugs right now going into clinical trials for senescent cells are antibodies that are engineered to have short lifespan, the antibodies. And normally antibodies have a tail so what do antibodies do? They bind the epitope, the foreign thing, and then they say to the immune cells, hey, I found something. And then the immune cells come, they latch onto the antibody, and then they eat it. I'm not going to eat the dog, of course. So in cancer, we want them to eat it. Here, if you want to have a few side effects, anyway, it works with these short-lived and immune suppressed antibodies. And also, what you need to do is just give a burst of killing. So the trucks kill the senescent cells, and then you have some time until the senescent cells replenish. It's very important to know that time, because if you give this PD-L1 inhibitor every week, it's like in cancer, you get autoimmune disease. But if you can give it once every 2 months or 3 months, less side effects. And how do we know? So again, we have our trucks model. It's a calibrated truck model. And we know from the truck model that in young people, trucks eat the senescent cells. Their half-life is days, but in old people, it's months. Why is it months? Because the trucks are so overloaded. And that gives you a time scale of when you need to do the treatment. In other words, the Truxton house model is useful for designing the clinical trial because you can simulate all these parameters and get it's like engineering. You can say, oh, let's make it, what's the optimal timing I need to give in terms of, but this is now going into the clinic. It's actually past, for Alzheimer, this passed stage 1, phase 1, which means it's not toxic in humans, and showed some efficacy. And now it's going into phase 2. So everything here is really, really young. In biology, young things tend to have a lot of hype, and then there's something else and something else. But Senolytics already around, I don't know, this idea 10 years or more, and it's my favorite anyway. More questions? Yeah.
You said earlier that when you have too many NK cells, or whatever that was, then it causes these autoimmune disorders. Is this only the case, would we only see this in young organisms that don't have enough damage to go around? Or is this also the case in people that have plenty of damage to be cleaned up, but it's still a problem if they have to?
So there's one disease, it's rare, but it's there, where old people have too much macrophages, and then they have a cytokine storm. That happens when a stem cell in the blood, so the bone marrow makes all the stem cells, all the blood cells. Old people increasingly have, sometimes 1 or 2 clones of stem cells takes over the whole bone marrow. So there's mutations happening. By the time you're, let's say, 60, you'll have 2,000 mutations in each stem cell. And if a certain stem cell has a growth advantage over time, it'll take over the bone marrow. So all the stem cells will be the same. And now it's a luck of the draw, what mutations they have. That's clonal homogeneity that appears in all old people. It's incredible. So this 100-year-olds, all their blood is made by basically a single stem cell clone with some random set of mutations that... Okay, that's something about old age. Also, your skin becomes...
Process take?
It's
around what point does it switch over from being a multitude to from being heterogenic?
Around 60, 70, you already start seeing this. 80, it's more common, and it's called the CHIP, clonal hematopoiesis of... I don't remember, it's something like that. Okay. If one of those mutations make this body make more macrophages, it's not so common, that creates a cytokine storm situation where a person dies from immune hyperactivity. So that's an example.
But like how and do these people that die of it, do they also have many senescent cells when they die, or do we see these people have...
That's not clear whether they have many senescent cells or not, because it's not standard to measure senescent cells in humans. Okay. It's in the lab.
What about in model animals then that have cytokine storms? Or do they not have them?
Okay, so model animals, if you remove the dagger of the NK cells, the number of senescent cells grows by so much that they die early, like progeria.
No, but I'm saying that. But wait, so all these additional macrophages, they're not actually killing senescent cells?
Oh, I see what you're saying.
That's what I'm asking. I'm asking, yeah,
Yeah.
do they kill you? Do they also kill the
I don't know. I mean, it's a good question to test, but the collateral damage is what kills you. So,
No, so
and only collateral damage. They produce inflammatory cytokines, and then they incite themselves to produce more cytokines, and that's called cytokine storm. And that makes all the cells in the body enter an alarm state where they're like, it's like hyper sick, and the macrophages also secrete reactive oxygen species. They're nasty, and that starts damaging tissues. And then there's another feedback loop. That's what happens also in COVID, in severe COVID. There's it's not the virus, it's the cytokine storm that kills the elderly people. So that's an example of, so what you're asking, do they
But just
kill?
having many, many macrophages without anything to do with that, that's not harmful in and of itself. The problem is that they keep reproducing it.
Yeah, I'm not an expert in this, and there's different flavors. Sometimes they also eat tissue. Sometimes they have. I just gave it as an example to say that there might be a limit to how many trucks it's good to have, right? Because you have a benefit and you have a cost, and sometimes the cost exceeds the benefit, especially if it's a non-linear process.
No, but then the question is like would you introduce more macrophages as the body gets older, as there's more damage to be soaked up?
Yeah, that's a good idea, but
As you're older, there's more
yeah.
damage to eat.
By the way, old people, if you look at the blood of old people, they have more macrophages and neutrophils at the expense of T cells and B cells. That's called myeloid shift. That's a major thing that happens in aging, also in pregnancy, but for different reasons. And the myeloid shift might be a response to what you're saying, but it's mild. So the body has some way to tune it. But I'm afraid of adding more macrophages. I don't know. I mean, it's just. I'm not sure. Anyway, what you're thinking is another way to enhance the trucks, right? That's the way. That's where you're going with it. Yeah. Yeah. So. Yeah.
Was the research into reducing the inflammatory signal from senescent cells?
Yeah.
And also, do they do any damage beyond the
Okay.
inflammation?
Right. So.
Because otherwise, they could just sit there.
Yeah. So this is the stink of the garbage. So inhibiting this is called xenomorphic. So there's a field also to make the senescent cells produce less of this or to neutralize it. So that's another whole approach. I don't know too much about it, but I know that it exists. It's like, I think it's like increasing X critical.
No.
It's not?
Like,
Because you need more garbage to kill you because each one is less stinky. Something like that. I don't know. I'm not sure. And then the same stuff they secrete, senescence-associated secreted profile, SASP, it also has a bystander effect where it turns normal cells into inflammatory cells. So it has a bystander effect that's when you do mathematical models, you can see that it can't be too big, otherwise these senescent cells will produce a wave that make your whole body senescent. So that's one of the things in biology. It exists, and you can make a whole career of the bystander effect without thinking about the mathematical magnitude of how important it is, right? Because you understand, right, that if a senescent cell makes two other cells senescent, and they'll make 4 cells senescent, it's going to create a senescence wave that makes your whole body senescent. So that effect can't be too strong. Did you explain myself? Yeah. So anyway, great questions. What else do you have in mind? Yeah.
If you have to guess, do you think there's someone alive today who will live to 150?
I think my kids live to 100 on average because of the 6 hours a day. I think there's no reason to think that'll stop unless we...
That's median. Max.
150 will require something like this. Will it happen? I don't know. I mean, I think the partial reprogramming... I mean, it's so hard to predict. What do you think? Think yes? I mean, again, there's people who will tell you yes. Scientists will say yes. If you just buy my supplement, alpha-ketoglutarate. So when you hear that, you should be wary. Okay, so the reality is it's basically going into the unknown. We just don't know. But I think one thing for sure, it's very creative to think of different solutions. You show so many different ways. It's just maybe the tip of the iceberg because the number of people working on this in biology is like the tip of the iceberg. And that's one reason I do this course. I wanna encourage you, if you think you can do something in this field after you see this course, maybe you can join it. And then you're riding on a large increase in funding for this field, which has grown by a factor of 10 in the last 10 years, and also the amount of philanthropy and government spending is growing like this. Social interest in this, the tools in our disposal. So it's probably one of the most exciting frontiers in science. Right at the early days. Of course, it's so nice on the blackboard. Of course, there will be problems, and then to overcome those problems, maybe you need a combination solution or many different solutions for different people. But it's fun to be there. Okay, it's time to say goodbye. We'll meet you for the last final party next week. Let's take a nice deep sigh of relief for a long, meaningful life.